some of the domestication events. Somewhat strikingly, perhaps surprisingly, they're clusters in time around 10,000 years ago. We do not really know why. Well, that was the time of the last glacial retreat, but then one could argue why would a glacial retreat require you to domesticate food? Maybe, I mean, there are all these wild-grown fruits and vegetables and roots and what have you growing around you. There's no more ice, so why not just harvest them? Still, for one reason or another, there's this peak of domestication during which a lot of different species of mammals and a lot of different species of crops, plant crops, have been domesticated nearly simultaneously and in a rather low number of different regions. So there were just a few regions in which most of the biodiversity has taken place. The fertile crescent in the Middle East, Southeastern Asia, the Mesoamerica and the Andean region, some regions in Africa. That's about it. And so what we can do now is we can look at the product of these thousands of years of selection by our own species, and we can do that using genetics, now that the genetics is at large scale is available to us, and we can try to find out what has actually happened over this last time, over this period of time. And this is simplified by the fact that for most of the species that were domesticated, their wild ancestors are still around. So this is the corn, and this is the teosinte, the wild-growing ancestor of contemporary corn, which is still around, still growing. And you can sequence the genome of one, the genome of the other, and try to find what is different, what are the genes, for example, that have changed most prominently. And you can also try to find regions in the genome and the gene variants, alleles that have experienced selection. And then speculate that perhaps that selection was happening in the course of domestication. And when we do that, we find a number of genes that actually do show the signals of past selection in the maize, but not in teosinte. The particular signal of selection that people have been looking at here is this signal of something called selective sweep, something we'll be talking about more later, whereby the polymorphism in a genomic region is reduced after selection. So you see in teosinte there is a high variability in this region, but just one of the different variants remains in maize, and that probably means that this single variant was better than all others. That's why it has spread. The particular gene we're looking at here is this TB1 gene, which is, and it's so this is a gene that corresponds to the structure of the lateral branching. So there are these large number of lateral branches in teosinte and just one central stem in the modern maize. And this thing is apparently better because it allows you to produce larger corn kernels. And the mutation that has happened here was apparently regulatory. It has occurred in a non-coding region, and it has regulated the transcription of this gene. And there was also another mutation which affects the gene TGA1, which is responsible for the loss of this hard kernel coating, turning it instead into these soft coats, which has, by contrast, happened in the coding region of the gene. It has been an amino acid So then one can ask questions like, how many genes are involved in the adaptation due to domestication? Were these mutations mainly in protein coding or in regulatory regions? How strong was the selection favoring this mutation? Did this selection act on new mutations that originated after the selection has been imposed? Or did it increase in frequency the variants that have already been there in the population before domestication? These are all important questions, and these are the questions that we really want answered for all the domesticated cultures, I would argue. So to answer them, we need a lot of sequencing data for both the ancestral and the domesticated populations. And the general patterns that you expect is something like this. The domestication, selection caused by domestication, reduces diversity. And it reduces diversity in particular in the genes that are actually that have this allele which improves your yield, which improves the trait that is actually selected for. So in the gene responsible for domestication, you expect to get this reduction in diversity and just have this variant that is sort of optimal. In some other genes may not be selected immediately, but may be selected later down the line, now that the modern breeds are formed, because these genes also confer some selective advantage, even if it's minor. The neutral genes, on the other hand, that do not do anything, are on average expected to preserve their variation, although the variation in these genes can also be lost due to their linkage with these other genes. So this is roughly what we expect. But so these are the questions. So what are the answers to these questions? Well, it turns out that in the case that people have studied, there are many genes involved, but just a few of these genes are, the usual pattern is that just a few of these genes are major effect genes, and then there are lots of genes with much smaller effects, affecting the same or other traits. So the first thing that happens in the domestication is this really strong selection favoring some major effect genes, such as, for example, the seeds. And then as you proceed with selection for yield, for example, then the varieties are better and better varieties are selected, and then the subsequent improvement is much more gradual. So that's the typical pattern. It does not have to be the case, but that's the typical pattern. The total number involved in such adaptation can be anywhere in the tens or in the hundreds. So these are some examples of the genes in maize, in rice, in tomato, in wheat. And you notice that the mutations can be both within the coding sequence of the gene or regulatory, or sometimes they can even be associated with the gene loss. So perhaps some genes are deleterious are advantageous in wild nature, but they are deleterious for the trait that we want to select, say, rice for. So in these genes, any mutation that would disrupt the activity of this gene would be positively selected in the domestication and crop breeding. So these are the genes that are selected early in the domestication process, and then there is a whole number of other genes that are involved in crop diversification that are being studied. You can also pinpoint the geographical location of the domestication event if you have enough data. And you do have enough data for the case of maize, in the case of origin of maize from Teosinte, because a lot of varieties of Teosinte are still growing here throughout Mexico, and you can compare the genomes of contemporary maize breeds to those of the Teosinte, and say, build an evolutionary tree, and you can see that these, so here the green, and so this is the same tree in two different formats, and the green and the red branches correspond to ancestral Teosinte specimen. Now this Oaxaca lineage is the earliest, best we can say, actual maize, not Teosinte lineage, that people have harvested from highland Mexico. That is the earliest one branching off, and then there is this whole diversity of maize varieties. So we're really actually fortunate, I would argue, that the different Teosinte breeds are still around, and people are now arguing more and more that it's very important to preserve the genetic variability in the ancestral lineages for the crops that we use. And that is important in terms of food security, because the contemporary crop breeds tend to be rather inbred and rather monomorphic, and being rather invariable makes you susceptible to different things, such as climate change or such as unexpected pathogen attack. And people are very concerned about the fact that we are now harvesting relatively few food crops, and so the whole well-being of the human, of humans depends on a rather low number of different genotypes of different crops. So it's really important to preserve the diversity and to study it more, and perhaps to make use of this diversity. And it's a really good thing that the Teosinte is still present there. One concern people have had about the genetically modified corn was that the genetically modified corn has been outnumbering the wild type varieties. People have claimed it's not. I have not been following the literature that much, but people have claimed that the genetically modified corn, which is resistant to herbicide, has outplaced the ancestral Teosinte varieties. If that claim is true, that would be a concern associated with the genetically modified organisms. Of course, all the people constantly tell you that GMOs are bad for you and are harmful medically and so forth. That is not true. They're much better tested than all the other things that we eat for centuries that no one has really cared to test for deleterious substances. But this ecological concern of GMOs outplacing the extant variety, the wild type varieties, would be a valid concern in this case. I don't know if it's true or not. I haven't looked at this literature recently. So here's a tomato, and again, in tomato, people know 6 major effect genes in which the substitutions occur, and then they make tomatoes more like the ones that now we get in the supermarkets, less like the ones that were ancestral. One of these loci, called FW2.2 for fruit weight, it increases, just linearly increases the fruit weight. Somewhat unfortunately, some of these advantageous genes are associated with deleterious traits that we do not want. For example, an allele in a gene that allows the tomatoes to be better stored in supermarket conditions is actually linked to another gene, just happens to be linked in the genome to another gene which makes the smell and the taste of tomatoes worse. There was a paper in Nature, I believe, last year, which actually points out that these 2 genes are associated. So it's not that this gene, which makes them be better kept in supermarket, has this side effect of reducing the nice taste and smell It is just that these two genes happen to be next to each other on one of the chromosomes. And therefore, if you select an allele in one of the genes, you automatically select the allele that you don't want on the other of those two genes. Now, this is a technical problem. These two, you can unlink those two alleles by breeding or by genetic modification of one type or another. So probably, hopefully, that will be solved, and people will be tomatoes will be having the taste they used to have apparently previously before this allele has spread. There is this lab trying to do exactly that. Their motto is "Make tomato great again," and so they're trying to unlink these two variants from each other. Selection for domestication can be extremely rapid. This is a nice experiment done in Novosibirsk in 1959, in which they attempted to domesticate the wild fox, and they selected it for friendliness to humans, and they just over a few generations made individuals that were also selected that were much more friendly to humans. Somewhat unexpectedly, the idea was to make nice fur-bearing foxes, wild silver foxes with nice fur that would also be friendly. But when they selected for friendliness, they automatically, unexpectedly selected for diminishing fur quality. So the fur became sort of dog-like, and also these foxes started having wavy tails and floppy ears, and they in general started looking more and more like dogs. So apparently there is some linkage, either pleomorphic or some genetic linkage between the traits that make you more dog-like and the traits that make you less aggressive. And so that was an unfortunate result, but anyway, that's a very interesting experiment genetically, and people are now actually doing the whole genome sequences of those foxes, trying to figure out what has actually happened. So this is a notorious example of domestication of pigeons, and Darwin was a big fan of pigeon breeds, and he writes that any ornithologist who would see all those different breeds would immediately say that these are of course different species. But we know they're not different species because we, with our own hands, have just bred them a few decades ago. So Darwin, this was one of the things that sort of led Darwin to believe that species are transmutable. And sometimes the species that have been domesticated a huge time ago, say 6,000 years ago, as is the case for chicken, they have been selected for these thousands of years, but then in the just a few decades ago, some new means of selection have been found, or selection has somehow been found to be more efficient ways to select were developed, and the artificial selection has accelerated rapidly. So people have been breeding chicken for thousands of years, but just over a century ago, people have produced this broilers lineage in which the meat content of a chicken grew radically, and also these layers breeds in which you get many more eggs. Right. So the dogs were once claimed to be the most ancient domesticated species, and that was based on mostly some fossil remains of dogs near human settlements dating back to about 40,000 years ago. But now if we look at the genetics of dogs, it seems that all the breeds, all the contemporary dog breeds have their common ancestor also around 10,000 years ago. So they were domesticated. The current domestication and the current diversity is traced back to about 10,000 years ago, and if there was a prior domestication event, it has left no trace in the current breeds, at least genetically. And you can again build a phylogeny of different dog breeds and see which dogs are sort of the oldest in that, which ones are the ones that have branched off first. And it turns out that this Basenji dog is the oldest dog breed. Have any of you guys met Basenji? You know Basenji? So a friend of mine has this dog. It's a fascinating dog. It doesn't bark at all, and it licks its paw and washes its face with its paw. So that it's very cat-like. It's pretty cute. Rather aggressive though. So it's a nice dog breed. So if you draw the evolutionary tree, so you'll see more of those trees. So get used to them. That's just a round. That's a usual tree drawn in this round shape. So you see that the blue color here corresponds to wolves, and then you can see that the first branch branching off after the wolves is the Basenji. And there is this whole diversity of different dog breeds, some of them more ancestral, some of them more derived. So it's a rather well-structured tree. By contrast, here is a tree of cat breeds, which is less structured in that all the domestic breeds are sort of mixed with the different wild breeds. And people have argued that this has to do with the, more with the fact that the cats are less tied to the humans and compared to dogs, and the cat can still not know whether it has been domesticated or not. It just comes into the house and then leaves the time it wants. Okay, so this is a fascinating example of goldfish, and it's fascinating in that it's also a case of domestication about 10,000 ago. It's a much more recent domestication. The striking thing is that it is extremely well traced. So you have all these different Chinese dynasties which have traced basically for the last millennium or millennium and a half what changes in the phenotype of the fish were observed at what point of time with the precision of a year. And that's exceptional. Apparently they had no better things to do, so that's an exceptionally detailed record of this evolution of this goldfish that we are in possession of. And so one can, compare this timeline with the genetics of the corresponding phenotypes, and it's pretty fascinating. So much of the variety of this fish is such that, first of all, it's striking, but also it's immediately obvious that much of this variety is completely unviable in the wild. Okay, so now none of these fish would do well in the wild if they were released. For example, this whole number of fish have no dorsal fin, and I'm actually not aware of any wild fish that can do without a dorsal fin. That's a very anatomically unnatural way for a fish to exist. So these guys would probably be eaten immediately outside of the aquarium. Okay, so there's a common carp, a similar story. So this is a guppy which were just first domesticated only in the early 20th century, and you see that there's this huge diversity of phenotypes achieved over several decades. Again, domestication and selection, artificial selection can be extremely efficient and can proceed extremely fast. Domestication comes with a cost. We sort of have hinted at that, right? The species that are domesticated are artificially selected to better suit our needs, also tend to lose their fitness if that fitness were measured in natural environment. And there are several reasons for that. So that's important. First of all, we can directly select for traits that are useless or even deleterious, such as the case with the dorsal fish or the goldfish, dorsal fin of the goldfish. We can select for useful trait states, such as, say, high milk yield, but we will simultaneously select for reduction of the overall fitness, because in the wild, the cow does not only care about high milk yield, it also cares about, say, better survivorship, better immunity, better ability to breed, and so forth. And so there are all these things are linked together, and there are trade-offs involved. So if you increase the milk yield indefinitely, you would perhaps reduce the immune function of this cow, making it more susceptible to disease in the wild, outside of the farm where you feed it antibiotics all the time and make it survive sort of artificially. If you strongly select for some trait, you also this can also lead to selection, sort of a collateral damage, selection for other traits that are linked to this selected trait, but are deleterious, something that you do not want. We will talk more about this, about the actual mechanistic genetics of this. This is something that we've seen in the example with the tomato smell, where the unwanted mutation raising frequency together with the wanted one. So that apparently happens pretty frequently. The selection in the captivity no longer cares about a large number of traits. If an individual lives in if a cow lives in a well-heated farm 12 months a year, the selection no longer cares about the cold production or something like that. So again, if this cow is released all of a sudden into the wild, it will not do well. And finally, in breeding and actually other causes of reduction of genetic diversity. So the genetic diversity goes down, as we've just discussed. So people have actually tried to find out how much of a problem that is, and here's one such study in which people have measured the ratio or the numbers of non-synonymous and synonymous substitutions on evolutionary lineages leading to wolves and comparing them to lineages leading to dogs. Now, if natural selection is efficient, its main function is conserving the status quo. It mostly wants the protein coding genes to encode the exact same proteins they have encoded for the last thousands of years. So, therefore, if natural selection is functioning, you expect the synonymous substitutions to happen at some rate, because selection doesn't care about them, but the non-synonymous substitutions to be comparatively rare compared to the synonymous ones. Okay? So selection tends to conserve the protein function and therefore the protein sequence. And we can see how well it does that. And you see that it does pretty well in the branches, in the evolution of the wolf, because this number of non-synonymous substitutions compared to synonymous substitutions is low. But it does not all that well in the lineages leading to contemporary dogs. So it means that these dogs are accumulating mutations, probably deleterious mutations, as we adapt them to, as they live in our flats, basically, without selection that would have prevented those mutations if they were living in the wild. Yes?
Wouldn't we expect the same picture if it was just a different selective pressure?
That's a good point. We would indeed expect an excess of non-synonymous substitutions under positive selection, and there are actually ways to distinguish between these two patterns, which we'll be talking about in detail. In this particular instance, there's a way to tell. I don't want to go into that now because there will be like half a lecture devoted to that later. But there is a way to tell that these are actually deleterious mutations as opposed to advantageous mutations. Well, we have still about 20 minutes, so let's start talking about, and perhaps we can even finish this whole section today. Let's talk briefly about the experimental evolution that people are doing. So this is the most famous experiment of experimental evolution. This is experiment done in Richard Lenski's lab in University in Michigan State, and it's called the long-term evolutionary experiment. It's the longest one in the running. It has been started in 1988, and actually Lensky has sort of never expected it to last that long, but it keeps producing very interesting results. They keep publishing Nature and Science papers out of it still all the time, and sort of it has predated, the start of the experiment has predated the onflux of the whole genome sequencing, and now Lensky was actually smart enough to store in the freezer, the ancestors of these evolving strains, and therefore we can now get them out of this fridge and sequence them and find the differences between the novel evolved lineages and these ancestral ones. So here's Richard Lensky, here's his student Rei Zahavi with a temple built out of the petri dishes that they use in the experiment. And so the experiment has been in the running since 1988, so it's what, 30 years, right? Yeah, 30 years, more than 30 years now. They started from a single ancestral strain, 12 independent lineages, I believe, and they have evolved for several tens of thousands of generations since. So what you can do, for example, now that you have genetic sequencing available is, so let me tell you what the environment was. These strains were living in an environment which was in a glucose-rich medium, so there was glucose there, but none of the additional substances that E. coli needs for growth. Those E. coli had to produce for themselves. And they would reseed the populations every 12 hours, I believe. So the ones that were selected were the ones most rapidly growing ones, to a large portion. And so here you have the schematic sequences of the genomes of E. coli, and going from the center to the outside of these circles is time. The one in the center is the most ancient one, just 2,000 generations from the origin of this, from the start of the experiment, and the one on the outside is the 20,000 generation. And you see, so the dashes here are the mutations, and you see that once a mutation arises, it usually stays put. Once mutation is observed in one generation, it is also observed in subsequent generations, although there are exceptions. Here mutation arises, then it is lost, and then it arises somewhere in the vicinity again. So that sort of hints that these mutations are adaptive, because otherwise they would have, if these were just accidental mutations you would observe in one of the sequences, you would not expect it to also survive in all the subsequent generations. And there are additional data hinting that these mutations are adaptive, and the most important of that is that we do see an excess of non-synonymous substitutions compared to synonymous ones. So as a matter of fact, all these mutations fixed during the first 20,000 generations were missing substitutions. substitutions, that is, they were non-synonymous ones. So of course, if you randomly throw mutations on a genetic sequence, by the way, what fraction of mutations, if you just randomly mutate a genetic sequence that is coding for a protein, how many of those mutations would you expect to be synonymous and how many would you expect to be non-synonymous? About, what is the ratio? What would the ratio be? 1 over 1 over 3. Well, which way?
Synonymous.
So one synonymous, one non-synonymous per 3. 33% of non-synonymous. Any other ideas? We should go back to the table and check it.
15%.
Sorry?
15% synonymous.
15% synonymous.
Yes.
Why 15?
Because we have about half of the amino acids that could be easily mutated in synonymous way, and only a third of the nucleotides should be mutated in the codon to be synonymous change, to give synonymous change, something like this.
Well, so the easy way to think about it is if you just think of some codon, say, I don't know, ATA. If you look at the table of genetic code, I don't have one on the slide, but what you normally see is that the mutation in the first position would be non-synonymous, and mutation in the second position would always be non-synonymous, and mutation in the third position would usually be synonymous, right? So if these would also be, if it was, the rule was that these are always non-synonymous, non-synonymous, synonymous, then 1/3 of the mutations would be synonymous, right? In effect, some of these are also non-synonymous, so it's more like 1 synonymous to 2.5 non-synonymous mutations. But roughly, you roughly expect 1 synonymous mutation per 3 non-synonymous mutations. Okay? By contrast, what they see is that all 26 mutations are non-synonymous. They sort of would expect 1/3 of those to be synonymous, right? They don't see that. They see all of them are non-synonymous, which hints at that they are advantageous. And so what they also can do is they can directly measure the fitness of these populations. How do they do that? Well, they take the current, the strain that has just evolved in the lab, and they go to the freezer, and they dig out of the freezer its great-great-great-grandmother. They mark one or the other in different ways, say with the antibiotic resistance gene, and they put them all in into a competitive assay in a single Petri dish, and they see how well they compete with each other, right? So they can, as a result, they can measure the fitness of the extant strain compared to another extant strain or compared to its ancestor. I think that's pretty cool. And they can see how the fitness of these strains increases with time. So you see that the mutations are accumulated at a rather uniform rate. However, the relative fitness increase is sort of slowing down a little bit. And the reason for that is that, so what it means probably is something that we saw when we discussed tomatoes and corn, is that the mutations to happen first are the mutations that have the strongest effect. So the most radical ones tend to happen early in this evolutionary experiment, and the ones happening later are the ones sort of fine-tuning the different processes, and they correspond to just small marginal increases in the fitness. You can go and see how much parallelism, how much parallel evolution you see. You have these different lineages, and you see that sometimes the exact same mutation has arisen independently in different lineages, which means that probably you just need one exact same mutation to achieve the adaptation that you want. Sometimes multiple mutations have arisen in the same gene, but these were not the identical mutations, but they probably had the identical result as they happened in the same gene. Importantly, many of the first, so what exactly function are these mutations functioning? Or what exactly do they do? Well, it turns out that the mutations that actually happen first are sort of boring. They, instead of improving functions of some genes, which is sort of see what adaptation is improvement, right? What they do instead is they turn off certain genes that the E. coli does not need in this very stable environment in which it finds itself. Indeed, as the glucose is constantly supplied to it, it does not need its own catabolism of glucose. And therefore it loses the different genes that are involved in glucose catabolism, and the glucose catabolic function goes down in time. And due to that, the E. coli saves on, say, genome replication, on, say, production of mRNA and production of the proteins of the enzymes that it doesn't need. Some other fascinating things that came out of those experiments, what they saw was that at some point in one of the experimental populations around generation 6000, what they saw was in fact a speciation event. They saw two lineages coming out of the same ancestral, and then evolution in these two lineages that originated in this single flask was sort of independent. One of the lineages accumulated adaptive substitutions independent of the other lineages. And one of the lineages sort of lived on the products of metabolism of the other lineage. That was what allowed them to coexist with one another. So I think that's a fascinating experiment. We will actually probably have one of the papers from the journal club devoted to that. So they have papers coming out all the time, and that's a hot thing that has happened in the field of evolutionary genetics recently. So here in the Lansky experiment, it seems that the fitness still grows, but it seems that it grows, it is slowing down. This growth is slowing down. And conceivably, perhaps in this stable environment, the E. coli will at some point of time reach the fitness peak, and then the evolution will, the increase in fitness, the adaptive evolution will stop. This does not seem to be the case in this evolution of a phage. Instead, people have evolved a phage in providing it with a naive influx of bacteria all the time, and the adaptation has not been observed to slow down. So this probably means that there is some conflict involved, which corresponds to this continuous arms race either between the phage and the host, or between the different phages co-infecting a single bacteria. So how well can we do if we impose artificial selection on some really simple model system, such as, for example, Drosophila? People have tried to play around with that, selecting for traits that seem to be unimportant, such as, for example, the number of bristles of small hairs on the abdomen and the thorax of the fruit flies. So if you see a fly and it's about to breed and it has a high number of these bristles, then you let it reproduce. If it has a low number of bristles, you destroy it. You can impose selection like that, and it turns out that people have been doing that for like a long time ago, in the mid-20th century, and it turns out that selection can be pretty efficient for some traits, and perhaps not as efficient for the other traits. For example, for this abdominal bristle number, you can easily reduce the number of the bristles, but you cannot increase them so easily. So here selection is not that efficient. By contrast with the thorax bristle number, you can increase them easily, but it's much harder to reduce them. So there are perhaps some conflicts involved, some trade-offs involved with bristle numbers. They are involved in some function that we understand not so well. So this is a fascinating long-term experiment in which people have been evolving maize. Again, maize and they started with the same lineage, and they have evolved it, produced different lineages out of it, and they have evolved it either to increase the content of the protein in the kernel or to decrease it. And you see they have, along the way, in this generation 46, they reversed the direction of the selection and then produced this reversal high protein lineage in which they started selecting for the reduced protein content in this lineage, which has already presumably lost some diversity right into this selection. So if it has lost all its diversity by this time, then you would not expect this green line to go all the way down to this original red line. By contrast, it does go down almost all the way down, which sort of means that there was still plenty of diversity here 50 generations later. You can also do the same with the oil content, and strikingly, it seems that you can put the oil content all the way down to 0, basically. You can produce just by this gradual selection, and you can see it's actually pretty gradual, the kernels that do not have any oil in them whatsoever. Finally, what you can do is to impose selection for a clearly deleterious trait, and this is an experiment that has been done by Mukai's lab about 50 years ago, and that is now being reproduced in our own lab, because it now makes sense to redo it in the era of genomic sequencing. And what you do here is so you can impose any kind of selection you want in an experiment, right? You're the boss, you can do anything you want. So you can deliberately select for a trait that will be exactly deleterious in the wild. So how about selecting for a reduced fetus? That's a perverse thing, but you can try to select the individuals that produce the low number of offspring, but kill those that produce large number of offspring, right? So the way you do it is you put together 2 fruit flies, and first they try having sex. If they have sex within 30 seconds after you put them together, which is what normally happens, you kill them both. If they wait for 5 minutes, then they survive and you allow them to breed. And then you increase the threshold, and then in the end you have these, stupidly looking males who just sit there and look at the female and don't do anything for some time, but eventually they do have to reproduce, otherwise you will not be able to select them, right? So like perhaps 15 minutes or half an hour later, they think, well, okay, I will produce. And then you can also kill the female if it produces 20 eggs. But if it produces just a couple of eggs or 5 eggs, then it survives. Okay? And it turns out that you can, by doing this type of selection, you can actually select for low sexual activity here. So this is the fraction of the males that did not mate for about 30 minutes, and in the end, so you see this number grew. So what we're now trying to do by trying to sequence those guys to see what were the genetic changes that actually happened in them, how variable they were, really, because it seems that these changes can be pretty variable. There are many ways to destroy the function, but we do not know whether that's true or not. So the reproduction of this is the work currently in progress. You can also artificially turn out selection entirely. Make populations in which there is no selection at all. And how do you turn off selection? You ensure equal rights to breed for everyone. If you do that, there's no selection by definition, right? Because selection is non-equal breeding. So you take a male and a female, allow them to breed, and they breed any number of offspring. But you just pick out one daughter and one son out of those offspring. Put them again together, and then allow them to breed. No matter how many offspring these parents have produced, you just keep one daughter and one son. This experiment has been initially called the middle class neighborhood, because because that's exactly what happens in the Protestant neighborhoods, middle-class neighborhoods in the United States, where you have all these nice small houses standing next to each other, and there's a white Anglo-Saxon Protestant family there who exactly have 2 children, always. And so, what happens then? Well, it turns out the fitness to the population goes down because the selection is basically turned off. So the relative competitive abilities of these fruit flies does diminish. That's about a decrease of about 1 or 2% per generation. If you do then try to measure the actual fitness of these flies under harsh conditions. And much of that is due to accumulation of new mutations that would have been selected against normally but are not selected against in this experiment. Okay, so that's it for today for the observations of evolution. So this was sort of a impressionistic, I would say, lecture that was aimed at giving you the impression of the different things that people have been doing, and also the different regularities in the evolutionary process that we observe. And next Monday, we will start by talking about the reconstruction of phylogenies